Up the down staircase. Cerebellum. The cerebellum consists of two lateral parts, the hemispheres, and the median part, the vermis. Numerous fissures divide the surface of the cerebellum into lobes, lobules, and gyri, which are termed folia. Anterior, posterior, and floccular-nodular lobes can be distinguished in the structure of the cerebellum, and according to its function it is divided into the largest subdivision, cerebrocerebellum, occupying most of the lateral parts of the hemispheres and receiving input from the cerebral cortex, spinocerebellum, which occupies the median and paramedian parts of the cerebellum, and vestibular cerebellum, which corresponds to the floccular-nodular lobe and receives information from vestibular nuclei. The surface of the cerebellum is covered with gray matter, the cortex, under which there is white matter. The long axis of most folia is transverse. Sections of the folia at right angles to their axis have a characteristic appearance called arbor vitae, which means tree of life. The gray matter is also represented by paired nuclei which lie in the depth of the white matter. The cerebellar cortex has three layers, namely the outer or molecular layer, the intermediate Purkinje cell layer or ganglionic layer, and the inner or granular layer. Purkinje cells are large neurons about 80 micrometers in diameter with pear-shaped soma. They are localized in the ganglionic layer, forming parallel rows. Their strongly branched dendrites are oriented across the folium, in each row all in one plane, and pass through the entire thickness of the molecular layer. The dendritic arbor is composed of a proximal domain or larger branches and a distal domain of spine-laden branchlets. In the longitudinal section of the folium, each dendritic arbor has the appearance of a cypress. Axons of Purkinje cells travel to the white matter of the cerebellum, where they synapse onto neurons of cerebellar nuclei. On their way, they give collaterals to other Purkinje cells, basket cells, and Lugaro cells. The Purkinje cell axons are the main output from the cerebellar cortex and exert an inhibitory influence on the cerebellar nuclei. Most output nerve fibers of the cerebellum originate from these nuclei. One exception is the floccular-nodular lobe, since its axons contribute to the vestibulo-ocular reflex and synapse directly on neurons of vestibular nuclei without passing through the cerebellar nuclei. That is why vestibular nuclei are considered displaced cerebellar nuclei. One of the inputs to Purkinje cells is clymin fibers, which originate from the inferior olivary complex. Each clymin fiber is intimately associated with only a single Purkinje cell, but establishes numerous synapses on it. These fibers are termed clymin because they climb along the Purkinje cell up to its dendritic branchlets. The climbing fibers exert an excitatory influence on the Purkinje cells. The ganglionic layer also contains candelabrum cells and Bergmann glia. Candelabrum cells are the most recently delineated distinct neuronal phenotype of the cerebellar cortex. Their pericardia are usually elongated along the vertical axis. They have 3 to 5 short dendrites which project into the granule cell layer and 1 or 2 long, rarely branched dendrites which ascend vertically into the molecular layer where parallel fibers provide afferent input to them. The axons seem to synapse onto Purkinje cell dendrites. Candelabrum cells are inhibitory GABAergic neurons. Basket cells have pyramidal or oval soma and are usually found in the inner third of the molecular layer. They have 4 to 10 straight thick dendrites forming a fan-shaped tree. The parallel nerve fibers cross the dendritic tree of both interneuron types at right angles, providing the excitatory synaptic input. Basket cell axons descend to the Purkinje cells and form collaterals that, before synapsing, envelop their soma like a basket, giving the cells their name. Some axons surround the initial axon segment of Purkinje cells, establishing exoaxonic synapse called pinceau, whereas the axons of stellate cells contact the dendrites of Purkinje cells. The granular layer contains several types of neurons, such as granule cells, Golgi cells, Lugaro cells, and unipolar brush cells. The granule cells are the smallest and the most numerous neurons in the brain. Their total number in the human cerebellum makes up about 75 to 80% of all the brain's neurons. Their unmyelinated axons run to the molecular layer and bifurcate in it in T-shaped manner into 2 horizontal branches, which run at right angles to Purkinje cell dendritic trees. These branches are called parallel nerve fibers because they run parallel to the surface of the cortex. The distance between the ends of the 2 branches of one fiber reaches about 6 millimeters. On their way, parallel fibers form about 100 synaptic contacts with Purkinje cell dendritic spines. Thus, one granule cell diverges to contact many Purkinje cells, wherein hundreds of granule cell axon synapses converge onto a single Purkinje cell. Granule cells are glutamatergic excitatory neurons. Mossy fibers are the second input to the cerebellum. They come from many sources, such as the cerebral cortex via pontine nuclei and pontocerebellar tract, the vestibular nerve and nuclei via vestibulocerebellar tracts, the spinal cord via spinocerebellar tract, and the reticular formation. Mossy fibers ascend into the cerebellum where they branch innervating granule cells in several cerebellar folia and giving collaterals to the basket and unipolar brush cells and neurons of cerebellar nuclei. Branches of the mossy fibers twist through the densely packed granule cells, ending with enlargements, the rosettes, which indicate synaptic contacts and give fibers a knotted appearance. That is why they are named mossy. Information from the mossy fibers to the granule cells is sent along the parallel fibers to the Purkinje cells for processing. The influence of the mossy fibers is excitatory. The mossy fiber rosettes, the granule cell dendrites, the Golgi cell terminals, and glial cells form cerebellar glomeruli. Each mossy fiber terminal contacts with dendrites of about 50 different granule cells. Each of them sends dendrites to different glomeruli. The glomeruli are wrapped by velate astrocytes, which are protoplasmic astrocytes with thin, veil-like processes that overlap each other. Glial ensheathment ensures structural support, electrophysiological insulation, and chemical maintenance of the interstitial fluid. As was said, cerebellar nuclei lie in the depth of the white matter and are usually called deep cerebellar nuclei. From lateral to medial, they are dentate or lateral nucleus of the cerebellum, emboliform, globose, and fastigial or medial nucleus of the cerebellum. Humans and some animals do not have distinct emboliform and globose nuclei, and this fused structure is called interposed or interpositus nucleus. The fastigial nucleus is located immediately over the roof of the fourth ventricle. Fastigial nucleus consists of diverse neurons heterogeneously distributed throughout the nucleus. Diameters of neuronal pericardia in the fastigial nucleus are 5 to 35 micrometers. Fastigial nucleus neurons are divided into projection neurons and interneurons, which have short axons connecting only with neurons within the fastigial nucleus. Fastigial nucleus neurons are glutamatergic, GABAergic, and glycinergic. The main afferent input to fastigial nucleus is inhibitory GABAergic Purkinje cell axons of the cerebellar vermis. As the vermis receives somatic sensory inputs from the head and proximal parts of the body via ascending spinal pathways, the fastigial nucleus represents a part of spina cerebellum. Fastigial nucleus also receives collaterals from mossy and clamin fibers. The fastigial nucleus sends its axons to the vestibular nuclei and the nuclei of the reticular formation, thus being one of the main outputs of the spina cerebellum, which modulates motor activity via vestibulospinal and reticulospinal tracts. The fastigial nucleus connects to the nuclei of the third, fourth, sixth, and seventh cranial nerves and nuclei of the medial longitudinal fasciculus, controlling head, facial, and ocular movements. There are also projections from the fastigial nucleus to the hypothalamus and limbic system. It indicates that fastigial nucleus is engaged in the regulation of visceral and emotional activities. The interposed nucleus is located between the fastigial and the dentate nuclei. Its cytoarchitectonics is similar to that of fastigial nucleus. Interposed nucleus receives inhibitory GABAergic Purkinje cell axons of the cerebellar paramedian or paravermal part, collaterals from mossy and climbing fibers, and nerve fibers coming as parts of cuneocerebellar and dorsal spinocerebellar tracts. Interposed nucleus sends output via the superior cerebellar peduncle to the contralateral red nucleus. The dentate nucleus is similar to the principal olivary nucleus. It also has a laminar structure and in the transverse section has crumpled bag-like appearance. Axons of dentate nucleus neurons leave it through the hilum and form dentatorubral and dentator-thalamic tracts. Dentate nucleus is the nucleus of cerebellum and therefore receives Purkinje cells axons from lateral parts of hemispheres. Dentate nucleus also receives collaterals from mossy and climbing fibers and nerve fibers coming from the cerebral cortex via pontine nuclei. Dentate nucleus is divided into two parts, namely the dorsal or motor domain and the ventral non-motor domain. Their projections are organized to distinct areas of the cerebral cortex via the ventrolateral thalamus. The dorsal part projects to the primary motor and premotor areas and takes part in the planning, initiation, and control of voluntary movements. The ventral part projects to prefrontal and posterior parietal areas of the cerebral cortex and responsible for cognition, language, and sensory functions. The use of immunostaining in several last decades demonstrated the presence of sagittal zones in the cerebellar cortex. Their best-known marker is aldolase C, glycolytic enzyme expressed in the Purkinje cells, also called zebrin-2. This name is due to the alternating reactive and non-reactive parasagittal stripes revealed by staining and reminiscent of the stripes on a zebra. The stripes are also generated by other markers, but all of them have the same general shape. They are oriented perpendicular to the cerebellar folds. They are narrow in the medial-lateral direction, but much more extended in the rostrocaudal direction. The Purkinje cells in each zone have connections with the particular cerebellar or vestibular nuclei, with the particular part of the inferior olivary complex, and are related to distinct functions. Cerebellar zones can be partitioned into several tens of microzones, which are arranged in long, narrow bands oriented perpendicular to the folia. Each microzone is presented by a group of the Purkinje cells having the same somatotopic receptive field. Microzones with their interconnected small parts of cerebellar nuclei and inferior olivary complex form the operational units, micromodules of the cerebellum. Interestingly, the olivary neurons generating climbing fibers belonging to the distinct microzone are usually coupled by gap junctions. This helps to synchronize Purkinje cells within a microzone on a millisecond time scale. Recently, the term cerebellar quantum has been proposed to define the smallest interconnected olivocorticonuclear entity. Thank you for attention.